There are rocks in northern Quebec that have been sitting in the same place for 4.3 billion years, older than the moon's current orbit, older than the first stable continents, older than almost anything else you could point to and call solid. And inside those rocks in the last decade, researchers found something that shouldn't have had time to exist yet: tiny tube-shaped structures, built from iron and carbon, in a pattern that on any younger rock would be considered an obvious, unremarkable trace of ancient bacteria. On a rock this old, it means something else. It means life may have already been here, quietly working, before the planet had even finished the most violent part of its own childhood. Pale blue drift. To understand why a handful of microscopic tubes in an obscure Canadian rock formation matters this much, you have to understand exactly how young, how raw, and how genuinely hostile the Earth still was at the moment those structures apparently formed. The planet itself only finished its main assembly around 4.5 billion years ago, gathering itself out of the same swirling disk of dust and debris that built the rest of the solar system. Shortly after that, in an event still considered one of the most violent moments in the story of any planet, a Mars-sized world collided directly with the young Earth, an impact so catastrophic it liquefied significant portions of both bodies, blasted an enormous ring of molten debris into orbit, and left behind the object we now call the Moon, condensing slowly out of that debris in the years and centuries that followed. For a long stretch afterward, Earth remained something closer to a slowly cooling wound than a planet. Its surface, for a period lasting many millions of years, is generally believed to have been either partially or entirely molten, an ocean of liquid rock rather than liquid water, radiating heat into space as it gradually solidified into the first thin, unstable crust. Even after that crust formed, the young Earth continued to be pummeled by leftover debris from the solar system's own formation, asteroids and comets slamming into the surface, with a frequency and violence almost impossible to picture from where you're resting right now. An era so relentless that some scientists have proposed it may have periodically sterilized the surface entirely, boiling away oceans and vaporizing rock again and again, resetting whatever fragile chemistry might have been trying to organize itself in the meantime. Given all of that, for most of the 20th century, the working assumption among scientists studying the origin of life was straightforward. Life could not have begun until this violence finally settled down, until the bombardment slowed enough for oceans to remain stable, crust to remain solid, and chemistry to have an actual, uninterrupted stretch of calm in which to slowly organize itself into something capable of replicating. That calm period was assumed to have arrived sometime around 3.8 to 3.5 billion years ago, and the oldest confirmed evidence of life, for decades, seemed to support that timeline closely. Fossils and chemical signatures clustering right around that same window, exactly when the story said they should. Then the evidence started getting older and older, and each time it did, the story had to quietly move its own starting line backward to accommodate what had actually been found. The structures discovered in Quebec, in a rock formation called the Nuvuagittuq Greenstone Belt, some of the oldest confirmed rock on the entire planet, pushed that starting line further back than almost anyone studying the field had seriously entertained before. The tiny iron-rich filaments and tubes found inside these rocks bear a striking resemblance to structures produced today by certain bacteria living around hydrothermal vents, deep-sea environments where mineral-rich, superheated water pours out of cracks in the ocean floor, creating narrow, localized pockets of chemical energy that some of the most primitive life on Earth still relies on today. If these ancient structures really do represent the fossilized remains of similar organisms, rather than some purely mineral process capable of mimicking biology by coincidence, it would place the origin of life at or before 4.28 billion years ago, a date so early it falls only slightly after Earth's crust had even finished its initial, unstable formation. And well within the window most researchers had assumed was still far too violent and far too unstable for anything alive to survive in, let alone begin. That word, if, is doing an enormous amount of careful, necessary work in that previous sentence, and it deserves to be treated with exactly the same seriousness researchers themselves have given it. Structures that resemble biology are not automatically evidence of biology, and the scientific community studying these particular rocks has spent the years since their discovery locked in genuinely careful, occasionally heated debate over whether what they're looking at is truly the fossilized trace of ancient microorganisms, or simply an unusually convincing mineral formation, produced by ordinary geochemistry, running through iron-rich fluids without any life involved at all. This distinction matters enormously, and it connects to a much older, much more painful lesson the field of early life research has already learned the hard way once before, from a different set of ancient rocks entirely. In the 1990s, a separate discovery in Greenland, in rocks dated to roughly 3.7 billion years old, was announced as potential evidence of some of the earliest life ever found, based on a specific chemical signature in the rock's carbon, a particular ratio between different carbon isotopes that living organisms, through the ordinary chemistry of metabolism, tend to leave behind in a distinctive, recognizable pattern. For years, this signature was treated by much of the field as some of the strongest available evidence for very early life, cited widely, taught in textbooks, held up as a landmark finding. Then, years later, a team of researchers went back to the exact same rocks with more sophisticated dating and analysis techniques, and found that the specific structures carrying that supposedly biological signature weren't structured the way early life's remains should be at all. They looked, on closer and more careful inspection, considerably more consistent with ordinary metamorphic processes, minerals shifting and recrystallizing under heat and pressure over billions of years, capable, it turned out, of producing a carbon signature that looked remarkably similar to a biological one without any life having been involved in creating it at all. That reversal, a landmark discovery quietly walked back after more careful scrutiny, became something of a foundational cautionary tale for the entire field of early life research, a reminder woven into how every subsequent claim of ancient biological evidence gets evaluated. It's precisely why the Quebec structures, despite their genuine promise, have not been universally accepted as confirmed evidence of life, and why the researchers who found them have been notably careful, in their own published work, to describe their finding using exactly the same cautious, hedged language potential biosignature, possible evidence, rather than the confident, declarative claim a discovery this significant might otherwise invite. Even the age of the Quebec rocks themselves remains genuinely, actively contested, in a way that illustrates just how difficult it is to pin down anything with real confidence this far back in Earth's history. The original research team, dating the rock using the decay of radioactive samarium into neodymium, an extremely long-lived isotope pair suited specifically to dating rocks this ancient, arrived at that striking figure of 4.28 billion years. A separate team of researchers, using a different radiometric dating method applied to different mineral samples collected from the same rock formation, arrived at a noticeably younger age, closer to 3.75 billion years, a full 500 million year disagreement that remains, even now, unresolved between the two research groups, each one confident in their own specific methodology, and each one publishing detailed technical rebuttals of the other's approach. That disagreement matters enormously for tonight's story, because it determines exactly how radical a claim we're actually dealing with. If the older date holds up, these structures would represent life existing shockingly close to the very moment Earth's crust first solidified, a timeline so tight it leaves barely any room at all for the kind of patient, gradual chemical experimentation most scientists assumed life would have needed before successfully organizing itself into something capable of replication. If the younger date is correct instead, the finding remains remarkable, still among the oldest potential evidence of life ever discovered, but comfortably within the time frame most existing models of life's origin had already anticipated, a genuinely important data point rather than a wholesale challenge to the entire established timeline. Both possibilities, notably, point in the same underlying direction, even if they disagree on exactly how dramatic that direction turns out to be. Every time researchers have pushed the search for the earliest traces of life further back in time, using increasingly careful, increasingly sophisticated methods, the evidence has kept obliging them, again and again, refusing to simply run out the way a truly rare, difficult, one-time chemical accident probably should. Life's fingerprints keep showing up earlier than expected, one discovery after another, stretched across 4 decades of gradually revised, gradually older starting dates. A pattern researchers in this field have started to treat as a signal in its own right, independent of any single contested rock formation. That pattern connects to something else worth understanding clearly, a broader, increasingly well-documented fact about life on this planet that reframes the entire question of when it started. Wherever researchers have gone looking for the absolute limits of where life can survive, deep beneath kilometers of solid rock, inside boiling acidic springs, frozen for millennia inside Antarctic ice, thriving in water so salty it would dissolve most ordinary cells on contact, life has, with almost frustrating consistency, already been there first. Microorganisms have been found living, actively, inside rock samples drilled from more than 3 kilometers beneath the ocean floor, in conditions of heat, pressure, and near total isolation from sunlight that would have seemed, to earlier generations of biologists, thoroughly incompatible with anything alive at all. Entire ecosystems exist around those same deep-sea hydrothermal vents we mentioned earlier, drawing their energy not from sunlight, the source every schoolchild is taught underlies essentially all life on Earth, but directly from the chemical energy of the vent's own mineral-rich, superheated water, an entirely separate, self-sufficient foundation for life that has almost nothing to do with the sun at all. This tenacity, this apparent willingness of life to colonize absolutely any environment offering even the faintest usable trickle of chemical energy, is precisely why so many researchers studying the origin of life have grown increasingly comfortable with the idea that it may have begun shockingly early, shockingly close to the moment conditions became survivable at all, rather than waiting patiently for calm, gentle circumstances that resemble the world we live in today. If life doesn't actually need calm circumstances, if it can take root in exactly the kind of violent, chemically extreme, geologically unstable environment the young Earth actually was, then there was far less reason for it to wait around for anything gentler before beginning. It could have started as early as the chemistry allowed, in exactly the kind of hostile, hydrothermal environment those Quebec rocks appear to preserve, and the 4 billion years since have simply been life quietly, patiently, continuing to do what it apparently started doing almost immediately. That single reframing, the idea that hostile conditions were never actually the obstacle everyone assumed they were, changes the shape of an entirely separate, equally strange mystery buried deep inside the genetic code of every living thing on Earth today. A mystery that reaches back toward something researchers have reconstructed with remarkable and genuinely unsettling precision, not from ancient rock at all, but from the DNA quietly sitting inside your own cells at this exact moment. That mystery has a name, one that sounds almost like something out of mythology rather than a genuine, carefully reconstructed scientific finding. Researchers call it LUCA, the last universal common ancestor, and unlike the contested rocks in Quebec, LUCA isn't a fossil at all. Nobody has ever found its remains, and given how ancient and how delicate the organism must have been, nobody ever expects to. Instead, LUCA has been reconstructed entirely from the inside out, pieced together not from stone, but from the shared genetic inheritance still quietly present in every living cell on the planet today, including the cells currently keeping you alive as you listen to this. The logic behind this reconstruction is simple to state, even though the actual execution took decades of painstaking, technically demanding work. Every organism alive today, from the bacteria in soil to the whale in the ocean to the person lying quietly in whatever room you're in right now, descends from a single common ancestral lineage, a lineage that, if you trace it back far enough, eventually converges into one single population, or in the strictest formal sense, one single kind of organism from which every subsequent branch of life on Earth eventually diverged. That convergence point is LUCA. It isn't the first living thing that ever existed, an important distinction worth holding onto. It's simply the most recent point at which every surviving lineage of life on Earth still shares a common origin, the last ancestor before the tree of life properly split into its major branches. Finding LUCA doesn't require digging through rock at all. It requires comparing genes, an enormous number of them, across an enormous number of different species, looking specifically for genes so fundamental, so essential to the basic business of being alive, that they've been preserved with only minor modification across every single branch of life for billions of years since. If a specific gene, or a specific biological function that gene performs, shows up consistently across bacteria and archaea, the two most ancient and most fundamentally different domains of life recognized by biologists, then that gene almost certainly existed already in the shared ancestor. Both domains eventually diverged from, rather than having been separately, independently invented twice by pure evolutionary coincidence. This method, called comparative genomics, has been applied with increasing sophistication over the past several decades, as the cost of sequencing an organism's entire genetic code has fallen dramatically, allowing researchers to compare thousands of complete genomes rather than the small handful available to early researchers working on this question in the 1990s. One of the most thorough and most widely discussed reconstructions of LUCA's genome, published by a research team working through an enormous comparative analysis of genes across thousands of modern bacterial and archaeal species, identified a specific set of genes, several hundred of them, so consistently, universally present across both of life's most ancient domains that they were judged very likely to have already existed in LUCA itself, inherited directly from that single, shared ancestral organism rather than evolved independently, twice, by chance. What that reconstructed genetic toolkit reveals about LUCA is genuinely remarkable and considerably more specific than most people expect a 4 billion-year-old, entirely hypothetical organism's biology to actually be. LUCA appears to have possessed genes for an ancient form of energy metabolism built around hydrogen gas, using it as a fundamental source of chemical energy, in a process closely related to one still used today by certain modern microorganisms living deep underground and around hydrothermal vents, organisms that derive their energy not from sunlight or oxygen at all, but directly from the chemical reaction between hydrogen and carbon dioxide. A pathway sometimes called the Wood-Ljungdahl pathway, one of the most ancient known routes by which living cells convert simple, inorganic chemicals into the energy needed to build and repair themselves. That specific detail, LUCA's apparent reliance on hydrogen chemistry rather than sunlight, connects directly, and rather beautifully, back to exactly the kind of hydrothermal vent environment those contested Quebec rocks appear to preserve. Modern hydrothermal vents, whether the volcanically superheated black smoker vents found along mid-ocean ridges, or the cooler, more chemically gentle alkaline vents like those studied extensively at a site called Lost City in the mid-Atlantic, continuously pump hydrogen-rich fluid up through networks of tiny, naturally occurring mineral channels, chemical gradients forming spontaneously between the vent fluid and the surrounding seawater. Gradients some researchers believe closely mimic the basic electrochemical setup every living cell still relies on today to generate usable energy. Protons flowing across a membrane from a region of high concentration to low concentration. Precisely the same fundamental mechanism, called chemiosmosis, that powers nearly every cell on Earth right now, including every single one of your own. This convergence, LUCA's reconstructed genetic toolkit pointing toward hydrogen-based, hydrothermal vent chemistry, and the physical, mineral structure of certain vent environments naturally producing exactly the kind of chemical gradient early life would have needed to get started, has become one of the most compelling and most widely supported modern theories for where and how life on Earth actually began. It's called the alkaline hydrothermal vent hypothesis, developed most prominently through the work of researchers studying sites like Lost City, and it proposes something genuinely elegant, that life didn't need to invent its most fundamental energy-generating machinery from scratch at all. It simply needed to learn how to capture and internalize a chemical gradient the Earth itself was already producing constantly, for free, entirely without any biology involved, inside naturally forming microscopic mineral chambers deep on the ocean floor. Picture for a moment exactly what these environments actually look like, because the specific geometry matters enormously to why this hypothesis has gained so much serious traction. Alkaline hydrothermal vents like Lost City don't erupt violently the way the more famous, blazing hot black smoker vents do. They form gently over centuries, as seawater reacts chemically with a specific type of mantle rock, producing towering, porous structures of carbonate mineral that can rise many stories above the ocean floor, riddled internally with an enormous, interconnected labyrinth of microscopic channels and chambers, each one only a fraction of a millimeter across. Warm, alkaline, hydrogen-rich fluid from deep within the Earth's crust flows continuously through this internal maze, meeting the cooler, more acidic seawater filling the surrounding ocean, and at the boundary between the two, across the thin mineral walls separating each tiny internal chamber from its neighbors, a natural, continuously renewing chemical gradient forms, entirely without any life present to create it. Some researchers studying the earliest stages of life's origin have proposed that these naturally forming, mineral-walled chambers may have functioned as a kind of pre-made, geological substitute for the cell membranes every living organism relies on today, containing and concentrating organic molecules within a stable, protected space, while the natural chemical gradient flowing across their thin mineral walls provided a continuously available source of usable energy, free of charge, requiring no biological invention whatsoever to access. Under this view, early chemistry wouldn't have needed to solve two of the hardest problems in life's origin, building a working membrane and generating usable energy, entirely on its own. It could have simply moved into structures the Earth had already built, using an energy source the Earth was already generating, and slowly, patiently, over an unknown span of time, adapted and refined the biological machinery needed to eventually leave those mineral walls behind and build its own membranes instead. Membranes made of the fatty, self-assembling molecules every modern cell continues to build itself from today. This hypothesis remains genuinely contested, and it's worth being honest about exactly how contested it still is, because it sits alongside several serious, competing ideas about where and how life actually began, each with its own dedicated community of researchers and its own body of supporting evidence. A separate, longer-standing tradition of research has focused instead on shallow, sunlit pools on the early Earth's surface, proposing that cycles of wetting and drying in these small, isolated pools could have concentrated organic molecules and driven the specific chemical reactions needed to build the first self-replicating molecules. An entirely different environment, entirely different chemistry, and entirely different implications for exactly where on the early Earth's surface. or beneath it, life actually first took hold. What makes the hydrothermal vent hypothesis particularly compelling to a growing number of researchers, though, is precisely the connection we've been building toward all evening. The fact that LUCA's reconstructed genetics point so specifically and so consistently toward hydrogen-based metabolism. The fact that this specific metabolic pathway is considered one of the most ancient known routes to cellular energy across the entire tree of life. And the fact that naturally occurring hydrothermal vent environments happen to provide, essentially for free, exactly the kind of chemical gradient this ancient metabolic pathway would have needed in order to get started in the first place. 3 independent lines of evidence: genetic reconstruction, biochemical pathway analysis, and geological environment, all quietly pointing toward the same general answer, is precisely the kind of convergence that tends to earn a hypothesis serious, sustained scientific attention, even while the full picture remains genuinely, honestly incomplete. There is something worth sitting with here, something that reaches beyond the specific mechanics of hydrogen gradients and mineral chambers. If this hypothesis holds up under continued scrutiny and continued fossil and genetic evidence, it means the deepest, most fundamental machinery keeping every living cell on this planet alive right now, the basic electrochemical trick of pushing protons across a membrane to generate usable energy, wasn't something biology cleverly invented on its own, through some long, patient process of trial and error. It was something biology borrowed, directly, from a chemical process the planet itself had already been running, quietly, for free, since long before anything alive existed to take advantage of it. Life, under this view, didn't so much begin as it learned to recognize, and eventually internalize, an energy source that had been patiently waiting in the dark long before it ever arrived. That single reframing changes how you might picture the entire early history of this planet, the 4 billion years or so stretching between the moon-forming collision and the world you know today. It's tempting, and it's how most of us were taught to picture it growing up, to imagine those early hundreds of millions of years as simply empty, a lifeless, violent prelude to be gotten through before the real story of biology eventually began. What the reconstructed genetics of LUCA, combined with the physical mineral reality of vents like Lost City, increasingly suggest instead is a planet that was, in its own slow, patient geochemical way, already quietly building the exact conditions life would eventually need, long before anything alive existed to take advantage of them. The same chemical gradients still forming today, in the same kind of mineral towers, on the same ocean floor, entirely without instruction, entirely without intention, simply because that is what happens when a particular kind of rock meets a particular kind of water under a particular kind of pressure, patiently, for billions of years, whether or not anything is ever there to notice. And something, eventually, 4 billion years ago or perhaps even earlier depending on which contested dating method for those Quebec rocks eventually wins out, did notice, in whatever the earliest, dimmest possible sense of noticing actually means for a simple chemical system slowly learning to sustain and replicate itself. It's worth going slower here, through exactly how researchers actually built this reconstruction of LUCA, because the methodology itself is one of the more quietly astonishing achievements in modern biology, a way of looking directly into the deep past without ever needing to find a single physical trace of what you're looking for. The foundational insight behind the entire technique goes back to a simple, almost obvious observation once you sit with it properly. Every gene in every living organism has a history, a lineage of its own, passed down from parent to offspring, occasionally altered by mutation, occasionally duplicated, occasionally lost entirely in one branch of the family tree while surviving intact in another. If you compare the same gene across enough different species, spanning enough different branches of life, you can reconstruct, with real statistical confidence, roughly what that gene's ancestral version must have looked like. The same basic logic historical linguists use to reconstruct long-dead, unwritten languages by comparing shared vocabulary and grammar across their modern descendants. Applying that logic to the entire genome of an organism, rather than a single gene, requires an enormous amount of computational effort, comparing thousands of individual genes across thousands of different species simultaneously, building what researchers call phylogenetic trees for each one. Essentially a family tree showing how that specific gene has branched and diverged across the entire history of life. And then cross-referencing all of those individual trees against each other to determine which genes were actually present together in the single ancestral organism sitting at the very root of the entire tree of life. It's a task that would have been utterly impossible before the era of cheap, widely available genome sequencing, and even now requires processing an amount of genetic data that would have seemed like pure science fiction to the researchers who first began speculating seriously about LUCA's existence back in the 1970s. The specific study that produced one of the most detailed and most widely discussed modern reconstructions of LUCA's genome examined nearly 2 million individual genes, drawn from thousands of different bacterial and archaeal species, searching specifically for genes that showed a very particular kind of evolutionary pattern, one consistent with having already been present at the root of the tree. In the ancestor, both major domains of life diverged from, rather than having evolved independently and separately within each domain after that split had already occurred. Out of that enormous initial pool, the analysis narrowed down to a specific set of genes, several hundred of them, meeting this exacting statistical standard, a genetic toolkit small enough to be genuinely surprising and specific enough to paint an actual, detailed picture of what kind of organism LUCA must have been, rather than simply confirming, in vague terms, that some kind of ancestor must have existed a very long time ago. That picture, once assembled, turned out to describe something considerably stranger and considerably more specialized than most people's intuitive mental image of the very first, most primitive possible life form. LUCA does not appear to have been some vague, generic, undifferentiated blob of simple chemistry, gradually and gently working its way toward complexity. The reconstructed genome describes something already surprisingly specific, an organism apparently adapted to live in a hot, oxygen-free, chemically extreme environment, deriving its energy specifically from the reaction between hydrogen gas and carbon dioxide, equipped with a functioning, if simplified, version of the genetic machinery every modern cell still relies on to read DNA and build proteins from it, and notably lacking several biological features that are essentially universal among organisms living in more ordinary, oxygen-rich, temperate environments today. That absence turns out to be almost as informative as the presence of any specific gene. LUCA's reconstructed genome shows no clear evidence of genes related to tolerating oxygen, a detail entirely consistent with everything else we know about the early Earth's atmosphere, which remained almost completely free of oxygen for well over 1 billion years after the planet first formed. Oxygen only beginning to accumulate in any meaningful quantity following the evolution of oxygen-producing photosynthesis, considerably later in the story of life on this planet. LUCA, in other words, lived in a world that would be lethal, or at minimum thoroughly hostile, to the overwhelming majority of the plants, animals, and even many of the bacteria alive on Earth today. A world without breathable air in any sense a modern organism would recognize, thriving instead in precisely the kind of hot, mineral-rich, oxygen-free, hydrogen-soaked environment those hydrothermal vent chambers we discussed a few minutes ago would have provided in abundance. There is a companion theory to all of this, one that approaches the same fundamental question. How did the first truly living, self-replicating system actually get started, from an entirely different angle, focused not on where life began geographically, but on what kind of molecule life actually needed to invent first. It's called the RNA world hypothesis, and it addresses a genuinely difficult puzzle sitting at the very heart of the origin of life question, a puzzle sometimes described, half-jokingly, as biology's version of the chicken and the egg. Modern life relies on a clean, elegant division of labor between two different types of molecule. DNA stores genetic information, a stable, durable archive of instructions, but it cannot, on its own, actually do anything with that information. It requires an enormous, complex team of specialized proteins to read it, copy it, and translate it into action. Proteins, meanwhile, are the workers, folding into intricate three-dimensional shapes capable of catalyzing chemical reactions, building structures, and carrying out essentially every active task inside a living cell. But proteins cannot build themselves from scratch or replicate themselves without detailed instructions supplied by DNA. Each of these two molecules depends entirely on the other already existing and already functioning correctly. Neither one, on its own, seems capable of getting the whole system started from nothing. The RNA world hypothesis proposes an elegant way out of this apparent paradox, built around a third type of molecule, RNA, chemically similar to DNA, but capable, unlike DNA, of also folding into complex three-dimensional shapes the way proteins do, meaning a single molecule of RNA could, at least in principle, both store genetic information and actively catalyze chemical reactions, performing both roles simultaneously rather than requiring a separate specialized molecule for each one. If the earliest form of life relied entirely on RNA, before DNA and proteins had ever evolved at all, a single self-replicating RNA molecule could, in theory, have solved the entire chicken and egg problem on its own, storing its own genetic blueprint while simultaneously acting as the chemical machinery needed to copy that blueprint and pass it on to the next generation. This hypothesis gained substantial, genuinely convincing support in the 1980s, when researchers discovered that certain naturally occurring RNA molecules inside modern cells are capable of catalyzing chemical reactions entirely on their own, without any assistance from protein enzymes at all, a discovery significant enough to earn its discoverers a share of the Nobel Prize in Chemistry. Even more tellingly, when researchers examined the ribosome, the enormously complex molecular machine every living cell relies on to actually build proteins by reading genetic instructions, they found that the single most critical, most chemically active part of the entire structure, the specific component that actually forms the chemical bonds linking amino acids together into a growing protein chain, is made not of protein at all, but of RNA. The ribosome, in other words, the single most fundamental piece of biological machinery connecting genetic information to actual functional biology, still carries, at its very core, a fossil of exactly the kind of RNA-based chemistry the RNA world hypothesis proposes life originally ran on entirely, billions of years before DNA and specialized proteins ever fully took over the roles RNA had apparently been handling largely on its own. Bringing these two threads together, the hydrothermal vent hypothesis describing where and through what energy source life may have first taken hold, and the RNA world hypothesis describing what kind of molecule may have first solved the fundamental problem of self-replication, gives researchers today a considerably more complete, if still genuinely incomplete, picture of life's earliest possible moments than existed even 2 decades ago. Picture once more those microscopic mineral chambers inside an ancient hydrothermal vent structure, warm, alkaline fluid flowing steadily through their walls, a natural chemical gradient forming continuously across each thin mineral partition. Within some of those countless tiny chambers, simple organic molecules, themselves capable of forming spontaneously under exactly these kinds of chemical conditions, may have slowly accumulated and concentrated, protected from the diluting effect of the open ocean by the chamber's own confining mineral walls. Among those accumulating molecules, by chance, driven by nothing more mysterious than ordinary chemistry given enough time and enough repetition, a molecule resembling early RNA may eventually have formed, one capable, purely by the accident of its particular folded shape, of helping to copy other similar molecules nearby, including, crucially, copies of itself. Once even a crude, inefficient version of self-replication became possible, however slowly, however error-prone, the entire subsequent story of evolution becomes, in principle, simply a matter of time and chemistry continuing to do what chemistry does, slight variations in each new copy, some copies happening to replicate slightly more efficiently than others, those more efficient variants gradually coming to dominate the local population within their protective mineral chamber. Generation after generation, an unimaginably slow, unimaginably patient process of chemical trial and error, refining itself continuously within the same kind of naturally forming, energy-providing structure the planet had already been quietly building for hundreds of millions of years before any of it began. That patient refinement, running for what may have been tens or hundreds of millions of years within those mineral chambers, is one plausible answer to a question that might otherwise seem impossible to address at all: how something as breathtakingly intricate as a functioning, self-replicating cell could ever have assembled itself from nothing more than ordinary chemistry and time. It didn't need to assemble all at once, in a single, impossibly improbable leap. It needed only to improve, incrementally, generation after generation, inside a stable, energy-rich environment patient enough to allow that slow improvement to continue uninterrupted for as long as it took. Exactly the kind of environment those alkaline hydrothermal vent structures appear, on the best current evidence, to have reliably provided. What makes this story feel less like ancient history and more like a living, ongoing presence is the fact that direct descendants of exactly this kind of ancient, hydrogen-breathing chemistry are still alive today, not as some rare, exotic curiosity confined to a single remote location, but as an entire, thriving domain of life that most people have simply never had reason to learn about. Archaea, one of the three fundamental domains biologists divide all life into, alongside bacteria, and the domain containing everything from mushrooms to whales to us, include numerous groups whose metabolism still closely resembles the ancient hydrogen-based chemistry reconstructed for LUCA, organisms called methanogens, which generate methane as a direct byproduct of combining hydrogen and carbon dioxide, essentially running, right now, largely unchanged across billions of years. Close to the exact same fundamental chemical reaction researchers believe powered the very first living cells on this planet. These methanogens are not confined to some single, isolated location either. They live inside the digestive tracts of cattle and termites, contributing to the methane those animals produce as a normal part of digestion. They live in the oxygen-starved mud at the bottom of swamps and rice paddies, in Arctic permafrost, and deep beneath the ocean floor, sometimes recovered from drilled sediment samples extracted from more than a kilometer below the seabed, cells that have apparently been surviving down there, metabolizing at an almost unimaginably slow pace, for potentially millions of years at a stretch, barely reproducing at all, simply persisting, patiently, in conditions of near total isolation from sunlight, from oxygen, and from any of the environmental change happening on the surface far above them. There is something worth sitting within the sheer, quiet persistence this represents, a detail that connects directly back to the earlier point about life's tenacity, but deepens it considerably once you actually understand the timescale involved. Some of the deep subsurface microbial communities researchers have studied appear to be reproducing so slowly, based on careful measurements of their metabolic activity, that a single generation, The time it takes for one of these cells to divide into two may stretch across centuries, or in some of the most extreme, most deeply buried examples studied, potentially thousands of years. These are organisms living lives so slow, so patient, that a single one of their lifetimes could easily outlast the entire span of recorded human history, sitting quietly in darkness, sustained by nothing more than a faint, continuous trickle of chemical energy seeping through rock, waiting. In whatever sense an organism without any nervous system at all can be said to wait, for absolutely nothing in particular. Simply continuing to exist because the basic chemistry of existing continues to be, however marginally, energetically favorable. That image of an entire hidden biosphere living beneath our feet at a pace so slow it barely resembles life as most of us understand the term, connects in a genuinely direct, unbroken line back to the reconstructed chemistry of LUCA itself, and by extension, potentially, back to whatever those contested structures in the Quebec rocks actually represent. If the alkaline hydrothermal vent hypothesis holds up, and if LUCA truly was a hydrogen metabolizing organism living in exactly this kind of hot, oxygen-free, mineral-rich environment, then the methanogens quietly persisting today in swamp mud and deep-sea sediment aren't simply distant, modified descendants of that ancient organism. In a meaningful biochemical sense, they are running something remarkably close to the very same metabolic program LUCA itself ran, largely unchanged, for the entire intervening span of nearly 4 billion years. A genuine, living connection to the earliest possible moment of biology on this planet, still quietly operating, right now, in environments most people will spend their entire lives never once considering. Researchers studying these deep biosphere organisms have begun to seriously grapple with just how much total biomass this hidden, subsurface world of life might actually represent. And the early estimates, while still carrying substantial uncertainty, are genuinely striking. Some calculations suggest the total mass of microbial life living beneath the Earth's surface and ocean floor, in rock and sediment far removed from sunlight, could rival, or in some estimates even exceed, the total mass of every plant and animal living on the surface combined, an entire, largely invisible biosphere operating on a completely different timescale and completely different energy source than the sunlit world most of us think of when we picture life on Earth. Hidden not because it is rare, but simply because it exists in places nobody had any obvious reason to look until fairly recently. This deep, slow, hydrogen-breathing biosphere offers something worth carrying forward as we move toward the closing thread of tonight's story, a genuinely different way of picturing what life on this planet actually is at its most fundamental level. The sunlit world of forests and oceans and animals, the version of life that built human civilization and every story we've ever told about what it means to be alive, may represent, in terms of sheer age and sheer persistence, something closer to a relatively recent, comparatively fragile innovation, a bright, fast-burning variation layered on top of a much older, much slower, much more patient foundation that has been quietly running, largely unnoticed, for nearly the entire history of this planet in the dark, beneath the rock, inside mineral chambers not unlike the ones that may have given rise to the very first version of it all, 4 billion years ago, before there was a moon in its current orbit, before there were continents, before there was almost anything else at all. There's a particular kind of quiet worth sitting with, in these final minutes of this thread, that has nothing to do with mineral chambers or comparative genomics or the precise dating of a rock formation in northern Quebec. It has to do with what it actually means to carry, inside every one of your own cells, a handful of genes so ancient, so deeply conserved, that they trace back, essentially unbroken, to that same original organism living in the dark 4 billion years ago. The machinery reading this sentence to you right now, whatever software converts these words into sound, runs on hardware, your own biological cells, that still carry fragments of code older than the moon's current orbit, older than every mountain range on the planet, older than nearly everything else that exists. That isn't poetic exaggeration. It's a literal, technical, genetically verifiable fact, sitting quietly inside every one of us, inherited across an unbroken chain of replication, stretching back further than most of us ever have real reason to consider. Something ancient really was already here, running quietly in the dark long before anything resembling a familiar planet existed, patiently working out the basic chemistry of staying alive, inside microscopic mineral chambers on an ocean floor that no longer exists in any recognizable form today. And whatever that something eventually became, stretched and reshaped across 4 billion years of relentless, patient, incremental change, it never actually stopped. It simply kept going, branch after branch after branch, until one of those branches eventually built eyes capable of looking backward, instruments capable of measuring backward, and a mind capable of wondering, quietly in the dark, exactly how far back the story actually goes. Something ancient, patient, and almost unbearably persistent had already found its way into those mineral chambers, already learned to draw energy from the gradient flowing quietly through their walls, long before there were continents in any shape you'd recognize today, long before the moon had settled into the stable, familiar orbit currently pulling gently at the tides of whatever ocean or lake happens to sit nearest to you tonight. Long before nearly everything else in the story of this planet had even begun. That question, how far back the story actually goes, turns out to have an answer considerably stranger than most people expect, because the deepest roots of this story may not actually begin on Earth at all. They may reach back further still, into the cold, dark space between forming stars, long before this planet had even finished condensing out of its own birth cloud of dust and gas. In September of 1969, a fireball streaked across the sky above the small town of Murchison in southeastern Australia, breaking apart with a sound loud enough to be heard for miles before scattering fragments of itself across the surrounding farmland. Local residents gathered pieces of the object over the following days, some of them found lying in fields still faintly warm, and handed them over to scientists eager to study what had fallen. What those scientists found, once they got the fragments into a laboratory, would go on to reshape an entire field of research, because the Murchison meteorite turned out to be carrying something nobody fully expected a rock from space to contain: an extraordinarily rich, complex mixture of organic chemistry, including more than 70 different amino acids, the same basic molecular building blocks every living cell on Earth uses to construct its proteins. What made this discovery so significant wasn't simply that amino acids were present. It was which amino acids were present, and in what specific mixture. Life on Earth builds its proteins from a very particular set of 20 amino acids, a specific, curated list out of a far larger number of amino acid variants that are chemically possible. The Murchison meteorite contained several of those same 20 amino acids used by terrestrial life, but it also contained dozens of additional amino acid variants that essentially never appear in any living organism on Earth today. A far broader, messier, more chemically diverse collection than biology actually uses. That specific pattern, some overlap with terrestrial biology, but considerably more diversity than biology ever bothered adopting, is exactly the signature researchers would expect from amino acids formed through ordinary, non-biological chemistry occurring somewhere out in space, rather than amino acids that had somehow been contaminated by earthly life after the meteorite landed. Researchers confirmed this extraterrestrial origin through several independent lines of evidence, perhaps most convincingly through the specific ratio of different carbon isotopes present in the meteorite's organic molecules, a ratio measurably different from anything found in terrestrial biology, and through the presence of amino acids showing what chemists call a racemic mixture. meaning roughly equal numbers of molecules built in each of the two possible mirror image configurations a given amino acid can take. Life on Earth, for reasons still not entirely understood, uses almost exclusively one of these two mirror image forms in its proteins, left-handed amino acids specifically, a strong, near-universal bias that any biological contamination would have reflected. The amino acids inside the Murchison meteorite showed no such bias, split roughly evenly between both mirror image forms, exactly the pattern you'd expect from amino acids formed through purely random, non-biological chemical processes occurring somewhere out in the cold vacuum of space, long before they ever had any contact with life on Earth at all. That single meteorite, more than half a century after it fell, remains one of the most extensively studied objects in the entire history of planetary science, and the picture it has helped build has only grown richer and more detailed as analytical techniques have continued to improve. Beyond amino acids, researchers studying Murchison and similar meteorites have identified sugars, including ribose, the same sugar molecule that forms part of the backbone of RNA, the ancient, self-replicating molecule we spent considerable time with earlier tonight. They've found nucleobases, the specific molecular components that pair together to encode genetic information in both RNA and DNA. They've found fatty acids, the same basic class of molecule modern cells used to build their protective outer membranes. In short, meteorites like Murchison appear to be carrying, scattered together in a single small rock, essentially every major category of molecular building block modern biology relies on, formed entirely through ordinary chemistry occurring somewhere out in space, long before any of it ever touched a living cell or a planet capable of hosting one. This finding forced an important, genuinely significant shift in how researchers think about the earliest stages of life's origin, one that connects directly and specifically back to everything we discussed earlier tonight about hydrothermal vents and LUCA's ancient chemistry. If simple organic molecules, including many of the exact building blocks life still uses today, are already being manufactured constantly, routinely, by ordinary chemical processes throughout space, then the earliest stages of chemical evolution on the young Earth may not have needed to start from complete scratch at all. The raw ingredients, quite plausibly, were already arriving from the very beginning, delivered continuously by the steady rain of comets, asteroids, and interplanetary dust that bombarded the young, still forming planet during its earliest hundreds of millions of years, seeding the surface and the oceans with a steady, ongoing supply of exactly the kind of organic chemistry those hydrothermal vent chambers would eventually need in order to get the process of chemical evolution started. Researchers have gone to considerable lengths to understand exactly where and how this space-based organic chemistry actually forms, and the answer reaches back further still, into environments considerably colder and considerably more remote than anything resembling a planet at all. Radio telescopes studying the enormous, cold clouds of gas and dust scattered throughout our own galaxy, the same kind of clouds that eventually collapse under their own gravity to form new stars and planetary systems, have detected an increasingly long and increasingly complex list of organic molecules already present, drifting quietly through interstellar space, in environments so cold, so diffuse, and so far removed from any star's warmth that most people would assume nothing chemically interesting could possibly be happening there at all. These interstellar clouds have been found to contain simple sugars, alcohols, and a wide range of increasingly complex carbon-based molecules, some of them containing more than a dozen atoms linked together in specific, recognizable structures, assembled not through anything resembling biology, but through an extraordinarily slow, patient form of chemistry occurring on the frozen surfaces of microscopic dust grains, tiny particles of rock and ice drifting through these clouds, coated in thin layers of frozen gas. radiation from nearby stars, along with cosmic rays streaming through the galaxy from far more distant, violent sources, provides just enough occasional energy to nudge simple molecules frozen onto these dust-grained surfaces into new, more complex configurations, one small chemical step at a time, across timescales of thousands or millions of years, far slower than any laboratory reaction on Earth. But given enough time, and interstellar clouds have an abundance of nothing if not time, capable of building up a genuinely impressive inventory of increasingly complex organic chemistry, entirely without a planet, a star, or any form of life anywhere nearby. When a cloud like this eventually begins to collapse under its own gravity, the first step in forming a new star and its surrounding planetary system, much of that painstakingly assembled organic chemistry doesn't simply vanish. A significant portion of it appears to survive the collapse, becoming incorporated directly into the icy, dusty disk of material that swirls around the young, forming star, the same disk that will eventually clump together into comets, asteroids, and planets. Under this view, when the young Earth was struck by a comet or an organic-rich asteroid during its own early, violent history, it wasn't simply receiving a random assortment of rock and ice. It was potentially receiving a delivery of pre-assembled organic chemistry, with a genealogy reaching back before the sun itself had even finished forming, molecules that had been slowly, patiently built on frozen dust grains drifting through interstellar space, incorporated into the material that built our entire solar system, and finally delivered, intact, to a young planet whose surface chemistry may have been more than ready to receive them. Recent space missions have added direct, physical confirmation to what had previously been inferred mostly through meteorite analysis and radio telescope observations of distant interstellar clouds. In the past several years, two separate spacecraft missions successfully traveled to individual asteroids, extracted small physical samples directly from their surface, and returned those samples safely to Earth for detailed laboratory analysis, a considerably more rigorous and more definitively uncontaminated method than analyzing meteorites that have already fallen through Earth's atmosphere and sat exposed on the ground before being collected. Samples returned from the asteroid Ryugu and separately from the asteroid Bennu both confirmed the presence of amino acids and other complex organic molecules, along with clear evidence of past liquid water having once been present within the asteroids themselves, adding direct, physical, laboratory confirmed weight to the broader picture meteorites like Murchison had already been suggesting for decades. This entire body of evidence, the meteorite analysis, the radio astronomy of interstellar clouds, and now the direct, physical sample return missions, converges on a genuinely striking reframing of exactly how far back the ancient roots of tonight's story actually reach. The organic chemistry needed to eventually build something like LUCA, working within those hydrothermal mineral chambers on the early Earth, may not have needed to assemble itself entirely from scratch, using only whatever chemistry happened to be locally available on the young planet's own surface. A meaningful portion of the necessary raw material appears to have already existed, already assembled, patiently built over unimaginable stretches of time in the cold dark between stars, before Earth itself, before the sun, before the moon-forming collision, before nearly everything else in this entire long, unfolding story. There is a particular, quiet vertigo worth sitting with here, one that reaches even further back than anything we discussed earlier tonight. It would already be remarkable enough to learn that the chemistry of life on this planet traces back nearly to the moment the planet's own crust first solidified. It is considerably stranger, and in its own way considerably more humbling, to learn that some of the actual physical raw material involved may predate the planet entirely, having formed on frozen dust grains drifting through a cold, dark molecular cloud long before our sun had even finished gathering itself together, carried along for the ride as that cloud slowly collapsed into a star and a swirling disk of planet-forming debris, delivered finally, patiently, across the last remaining stretch of that journey, to a small, rocky world circling a completely unremarkable ordinary star, where it apparently found, in a scattering of microscopic mineral chambers deep on an early ocean floor, exactly the right conditions to finally, after everything, become something that could reproduce and grow, and eventually, 4 billion years later, wonder about where it had actually come from. None of this displaces or contradicts anything we discussed earlier this evening about hydrothermal vents, or about LUCA's reconstructed genome, or about the ongoing, unresolved debate over the age and biological status of those structures in the Quebec rocks. It simply adds another layer underneath all of it, pushing the true starting point of tonight's story back further than the planet itself, connecting the earliest chemistry of life on Earth directly to the cold, patient, largely invisible chemistry constantly occurring throughout the wider galaxy, in molecular clouds that are, even now, quietly building up their own inventory of organic molecules, waiting, in whatever sense chemistry can be said to wait, for a star and a planet capable of eventually receiving them. It's worth pausing here to consider what this reframing actually implies for the broader question of whether life exists anywhere else at all, a question that has run quietly underneath nearly everything discussed across this entire evening's journey through ancient rock and reconstructed genomes and interstellar chemistry. If the raw organic building blocks life requires are genuinely common, manufactured routinely by ordinary, well-understood chemical processes occurring throughout the galaxy, in every collapsing molecular cloud that ever gives rise to a new star and planetary system, then the specific ingredients needed to eventually build something like LUCA may not be rare or exceptional at all. They may be, in a very real sense, a routine, near-universal byproduct of how stars and planets form in the first place, delivered as a matter of course to essentially every rocky world capable of receiving cometary and asteroidal impacts during its own early history, which describes, as far as anyone can currently tell, the overwhelming majority of rocky planets known to exist anywhere in the galaxy. Under this view, the genuinely rare, genuinely difficult step in the entire story may not be manufacturing the necessary organic chemistry at all, since the universe already appears to be doing that routinely, almost effortlessly, on an ongoing basis throughout its own vast, cold, patient interstellar chemistry. The rare step, if there truly is one, may instead be everything that happens after those ingredients arrive. The specific, difficult, still poorly understood transition from a rich mixture of organic molecules into something capable of actually organizing itself into a self-replicating system. The exact transition those alkaline hydrothermal vent chambers may have provided the necessary environment for here on Earth 4 billion years ago, and which may or may not have found a similarly suitable environment anywhere else in this galaxy of hundreds of billions of stars, each one surrounded in all likelihood by its own quiet, patient supply of the same basic organic ingredients, waiting, as they waited here for hundreds of millions of years, for the right combination of chemistry, energy, and time to finally turn them into something more than simply the raw material for a story that might, or might not, ever actually begin. There is a more radical version of this same basic idea, one that pushes past simply delivering the raw ingredients for life and asks whether something considerably more advanced, life itself, fully formed and already self-replicating, might have made the same interplanetary journey intact. Researchers call this broader family of ideas panspermia, and it exists today in several distinct, carefully separated versions, each carrying very different levels of scientific support, and it's worth being honest and precise about exactly which version the evidence actually favors, because popular retellings of this idea frequently blur together possibilities that serious researchers treat as fundamentally, importantly different from one another. The most modest and by far the most scientifically supported version, sometimes called molecular panspermia, is essentially everything we've just spent the last several minutes discussing: the delivery of organic building blocks, amino acids, sugars, nucleobases, rather than living organisms themselves. This version requires no exotic new biology and rests entirely on well-documented, repeatedly confirmed chemistry, exactly the kind of evidence Murchison and the more recent asteroid sample return missions have provided directly. A more ambitious version, sometimes called lithopanspermia, proposes something considerably harder to confirm: that actual living microorganisms, rather than simply organic chemistry, could survive the extraordinarily hostile journey between planets, embedded inside rock ejected from one world's surface by a sufficiently powerful asteroid impact, drifting through the vacuum of space for potentially thousands or millions of years, before eventually landing on another world and, if conditions there happen to be suitable, resuming life exactly where it left off. This version requires organisms capable of surviving an extraordinary combination of hazards simultaneously: the violent shock of an impact powerful enough to launch rock entirely off a planet's surface, the extreme cold and near total vacuum of interplanetary space, and prolonged exposure to cosmic radiation unfiltered by any planetary atmosphere, sustained potentially for an enormous span of time before ever reaching a new world capable of supporting it. Remarkably, this version of panspermia is not quite as far-fetched as it might initially sound, because researchers have directly tested several of its individual required steps under laboratory conditions, and in some cases in actual orbital experiments, with results that have genuinely surprised many scientists in the field. Certain hardy microorganisms, particularly certain species of bacteria capable of forming a dormant, highly resistant spore state, have been shown to survive laboratory-simulated impact shocks of a magnitude comparable to what a real asteroid impact would generate. Separately, samples of dormant bacterial spores and even certain small, multicellular organisms called tardigrades have been deliberately exposed to the actual vacuum and radiation environment of open space, attached to the exterior of orbiting spacecraft for extended periods, and a meaningful fraction of these organisms have been recovered afterward still viable, capable of resuming normal biological activity once returned to more hospitable conditions. None of these individual experiments proves that lithopanspermia has actually occurred, either delivering life to Earth from somewhere else, or potentially carrying Earth's own life outward to other worlds following a major impact event in our own planet's history, a possibility researchers have specifically raised regarding debris known to have been ejected from Mars toward Earth, and in principle, capable of traveling in the opposite direction, as well during earlier periods in the solar system's history. What these experiments do establish, with real, direct, physical evidence rather than pure speculation, is that the individual physical hurdles this more radical version of panspermia would require, an organism surviving violent ejection, prolonged exposure to vacuum and radiation, and eventual survivable arrival, are not obviously impossible, even if the full, complete journey from one planet's surface to another's remains, for now, entirely unconfirmed and genuinely difficult to test directly. Where the current, honest scientific consensus actually sits, after all of this research, is considerably more modest and more carefully bounded than either the most dramatic popular version of panspermia or the most dismissive rejection of the entire idea. Molecular panspermia, the steady, ongoing delivery of organic building blocks from space, is essentially settled fact at this point, confirmed repeatedly through multiple independent lines of evidence stretching from Murchison in 1969 through the direct sample return missions of just the past several years. Lithopanspermia, the transport of actual living organisms between worlds, remains a serious, actively studied hypothesis, with individually tested components lending it genuine plausibility, but without any direct evidence yet confirming that the complete journey has actually occurred, either delivering life to Earth or carrying it elsewhere. What both versions of this idea share, regardless of exactly how far the actual transport of biological material between worlds ever really went, is a fundamental reframing of how connected the origin of life on this one small planet may actually be to the wider chemistry and physics of the entire galaxy surrounding it. The story we began this evening with, a handful of contested, ambiguous structures inside 4 billion-year-old rock in northern Quebec, no longer sits isolated, a strange local event confined entirely to this one particular planet's own particular geological history. It sits instead at the very end of an extraordinarily long, patient chain of chemistry, reaching back through the earliest violent bombardment of the young Earth, through the disk of dust and debris that built our entire solar system, and further still, into the cold, dark, slowly evolving chemistry of the molecular cloud our sun itself was born from. A chain of cause and consequence stretching back considerably further than the planet we're standing on, considerably further than the star that planet orbits, reaching almost, in its own quiet, patient way, back toward the very beginning of everything. There's a particular image worth carrying forward from this thread before it settles, something to hold alongside the mineral chambers and reconstructed genomes from earlier tonight. Picture a molecular cloud, vast and cold, drifting quietly through the galaxy for millions of years before anything within it ever begins to collapse into a star. Inside that cloud, invisible to any eye on the frozen surfaces of countless microscopic dust grains, chemistry is already patiently at work, one slow reaction at a time, building sugars, amino acids, the scattered early ingredients of biology, entirely without purpose, entirely without direction, simply because that is what happens to certain simple molecules given cold, time, and the occasional nudge of stray radiation passing through. That cloud has no way of knowing whether any of what it's building will ever matter to anyone or anything. It simply continues, the way it always has, the way similar clouds are continuing right now, scattered throughout the galaxy, quietly assembling the same basic chemistry, waiting for whichever of them eventually happens to collapse into a star, and whichever fragment of dust and ice eventually happens to become a planet close enough, warm enough, and stable enough to receive what has already patiently been prepared for it. That patient preparation reaches back further than nearly anything else discussed tonight, and it leaves an entire, still open thread waiting on the other side of it. The question of what happened after those ingredients actually arrived, after they settled into their own mineral chambers on the young Earth's ocean floor, and slowly, across an unknown but almost certainly enormous span of time, began the long, uncertain, still not fully understood process of turning simple chemistry into something capable of calling itself alive. That process, the slow, patient transformation of delivered chemistry into something capable of replicating itself, is where every thread we've followed tonight finally converges, and it's worth walking through exactly how these pieces fit together, one final time, before letting the whole shape of the story settle. Picture the young Earth, somewhere in that narrow, hard-to-pin-down window between 4.5 and 4 billion years ago, its crust freshly solidified, still absorbing the last heavy pulses of the bombardment that had shaped its earliest history. Comets and organic-rich asteroids, carrying chemistry assembled patiently across the cold, dark chemistry of the solar system's own birth cloud, continue delivering their cargo of amino acids, sugars, and nucleobases to the planet's surface and oceans, adding to whatever similar chemistry may have already been forming locally, through Earth's own early atmospheric and oceanic chemistry. Deep beneath the young ocean's surface, along volcanically active ridges where fresh mantle rock meets seawater, alkaline hydrothermal vent structures begin forming, their interiors riddled with microscopic mineral chambers, warm, hydrogen-rich fluid flowing continuously through their walls, a natural, ceaseless chemical gradient forming at every thin partition, separating one tiny chamber from its neighbor. Somewhere within that vast, natural laboratory, chemistry delivered from space met chemistry generated locally by the planet's own restless geology, concentrated and protected within the confining walls of countless microscopic mineral chambers, and began, slowly, patiently, across a span of time that may have stretched for tens or hundreds of millions of years, the long, uncertain, and still not entirely understood process of self-organization, molecules interacting, occasionally forming crude, imperfect copies of each other, those copies competing, however feebly, for the same limited pool of raw material and available energy, the earliest, dimmest possible echo of what would eventually become natural selection, running not inside anything resembling a modern cell yet, but simply within the boundaries of a naturally occurring mineral chamber, doing the work a cell membrane would eventually take over entirely on its own. Out of that long, patient, largely invisible process, something eventually emerged capable of maintaining and replicating itself reliably enough, consistently enough, to be called, by any reasonable definition available to modern biology, alive. It would have been almost nothing like the life you're familiar with, no visible shape, no motion anyone could have observed, nothing resembling a cell as we'd recognize one today, likely little more than a self-sustaining, self-replicating chemical process quietly running within its mineral shelter, invisible, silent, and utterly indifferent to the 4 billion years of consequence its continued existence was about to set in motion. That process eventually refined itself into something recognizable as a true cell, developing its own membrane, its own genetic storage system, its own metabolic machinery, eventually becoming the organism researchers have spent decades painstakingly reconstructing through comparative genomics. The shared ancestor whose genetic fingerprint still survives, quietly, in every living thing on the planet today, LUCA. Somewhere between that reconstructed organism and the world you're sitting in right now, life diverged, again and again, into every subsequent branch of its own family tree, into bacteria and archaea first, and eventually, considerably later, following the same kind of rare cellular merger we discussed earlier, into the entirely separate branch of complex life that would eventually include every plant, every fungus, every animal, and considerably more recently still, a species capable of building instruments sensitive enough to detect 4 billion-year-old chemical fingerprints in ancient rock, sequencing technology sophisticated enough to reconstruct an organism nobody has ever actually seen, and spacecraft capable of traveling to distant asteroids and bringing pieces of them home, specifically to confirm, directly and physically, exactly the kind of interstellar chemistry theorized decades earlier from meteorite fragments and radio telescope observations of distant, cold, patient clouds. There is something worth sitting within the sheer, almost unbearable scale of patience this entire story represents. Patience measured not in years or decades, but in hundreds of millions of them. Patience belonging not to any single organism, since nothing alive during any single stretch of this process could have possessed anything resembling awareness or intention, but to the process itself, to chemistry simply continuing to do what chemistry does, given enough time, enough energy, and enough stable, protective structure to keep doing it uninterrupted. Nothing about this story required guidance, or purpose, or any external hand shaping its outcome. It required only the raw materials, largely delivered from space, the right kind of energetic environment, generously provided by the planet's own ordinary geology, and time, an amount of time so vast it borders on genuinely difficult to hold in the mind for more than a few seconds before your attention naturally slides back towards something more immediately graspable. And yet here you are, at the very end of that entire process, close enough to it now that you can lie quietly in a room somewhere, listening to a description of your own deepest origin, considerably deeper than your parents', deeper than any named ancestor in any family record, deeper than any species you evolved from, deeper even than the first true cell that ever managed to hold itself together, reaching all the way back to raw chemistry drifting through a cold, dark cloud of dust and gas, patiently assembling itself over millions of years before our sun had even finished forming, before there was an Earth for it to eventually land on, before there was anyone anywhere capable of caring whether any of it happened at all. Something ancient really was here before us, in every sense the phrase can honestly carry. Ancient in the sense of those contested structures in Quebec, potentially older than nearly any other confirmed trace of life on the entire planet. Ancient in the sense of LUCA's reconstructed genome, carrying chemistry still recognizably, functionally present in the microorganisms quietly persisting today in swamp mud and deep-sea sediment, and the digestive tracts of grazing cattle. And ancient in a sense reaching further back still, older than the planet itself, chemistry patiently built on frozen dust grains drifting through the space between forming stars, carried along for the ride as our own solar system slowly gathered itself together, delivered finally, across the last remaining leg of an almost unimaginably long journey, to a small, rocky, unremarkable world that happened, against every reasonable expectation, to have exactly the right conditions waiting to receive it. None of this makes the story any less strange, and it's worth resisting the temptation, here at the end, to tie it into a neater, more comfortable bow than the actual evidence allows. The age of the Quebec rocks remains genuinely, actively contested between research teams who have not yet resolved their disagreement. The precise details of LUCA's environment and metabolism remain a reconstruction, built from careful, rigorous, but ultimately indirect genetic evidence, rather than a discovered fossil anyone has ever actually held in their hand. The exact mechanism by which delivered organic chemistry first organized itself into something capable of true replication remains one of the most significant open questions in all of science, actively researched in laboratories around the world, without any single, universally accepted answer yet available. What is settled, genuinely and repeatedly confirmed across multiple independent lines of evidence, is simply the outline, the shape of the story rather than every one of its finest details, chemistry arriving from space, an energetic, protective environment provided by the young Earth's own geology, and an almost inconceivably long stretch of patient time, somehow, eventually, producing everything that followed. There is a reason researchers studying this question devote entire careers to it, beyond simple curiosity about our own planet's distant past. Every piece of this puzzle, painstakingly assembled here on Earth, doubles as a kind of instruction manual for evaluating the chances of life existing anywhere else. If the ingredients truly are common, manufactured routinely throughout the galaxy by ordinary interstellar chemistry, and if hydrothermal environments capable of hosting the kind of protected, energy-rich chemistry LUCA apparently relied on are similarly common among rocky worlds generally, then the specific, local conditions that mattered here on Earth may not have needed to be nearly as rare or fortunate as earlier generations of scientists once assumed. This is precisely why the same researchers studying ancient Earth rock formations and reconstructed ancestral genomes are so often the same researchers paying close attention to news of a distant exoplanet's atmosphere, or a subsurface ocean detected beneath the icy crust of a moon orbiting Jupiter or Saturn, worlds like Europa and Enceladus, both suspected to harbor exactly the kind of hydrothermal activity and exactly the kind of chemical energy gradient that may have gotten life started here in the first place, sealed away beneath miles of ice, entirely hidden from sunlight in conditions that would have seemed, to any earlier generation of scientists, thoroughly unpromising, right up until researchers understood just how little life on Earth itself may have ever actually needed sunlight to begin with. That connection threads directly back through everything discussed across this whole evening, and through other evenings we've spent together exploring distant worlds and their own possible chemical signatures of life. The same basic reasoning that makes a molecule like dimethyl sulfide detected in a distant exoplanet's atmosphere worth taking seriously. The same reasoning that makes a moon like Europa worth the enormous expense and difficulty of a dedicated spacecraft mission traces back to lessons learned first and most thoroughly right here, in the deep, patient history of our own planet, in mineral chambers on an ocean floor that no longer exists, in genes reconstructed from creatures too small and too ancient to ever leave a proper fossil, in amino acids extracted from a rock that fell from the sky above a small Australian town more than half a century ago. Every one of those threads, studied separately by researchers working in entirely different specialties, quietly reinforces the same underlying picture, that life, wherever and whenever it manages to begin, may not require anything especially exotic or improbable at all, simply the patient, ordinary chemistry the universe already appears to be running constantly, given enough time, enough energy, and the right kind of sheltered, protected space in which to keep working, uninterrupted. For as long as it takes. That outline is enough, on its own, to change how a quiet night like this one might actually feel. The next time you happen to be standing outside after dark, looking up at whatever portion of the sky happens to be visible from wherever you live, it's worth remembering that the same basic chemistry currently keeping you alive, the amino acids in your proteins, the fatty molecules in your cell membranes, quite possibly trace their earliest origin to exactly that kind of dark, star-filled sky, assembled slowly, patiently, on frozen dust grains drifting between the stars long before any of those stars, including our own, had even finished forming. You are not simply standing beneath the universe, looking outward at something separate from yourself. In a very real, very literal, chemically verifiable sense. A meaningful portion of what makes up your own body was already out there first, waiting patiently in the cold and the dark, for a very long time before it ever found its way home. There's a specific kind of comfort available in that fact, one worth sitting with directly before we let tonight close. It would be easy for a story spanning 4 billion years, reaching back before the planet itself, to leave you feeling small in an uncomfortable way, a brief, fleeting presence at the very tail end of a process too vast and too patient to have ever had any particular concern for whether you, specifically, ever came to exist at all. That's true, in its own narrow sense. But it isn't the whole picture, and it isn't, honestly, the part of the story that tends to stay with people once they've actually sat with it for a while. What tends to stay instead is something closer to genuine kinship, a recognition that you are not separate from any of this, watching it unfold from some safe distance outside the story. You are the story, or at least the current, temporary shape it happens to be wearing right now. 4 billion years of patient chemistry finally organized into something capable of understanding, in real and specific detail, exactly how it got here. Every atom of carbon in your body was forged inside a star that died before the sun was born. Every amino acid your cells still rely on carries a chemical lineage reaching back into the same cold, dark interstellar clouds where Murchison's rich mixture of prebiotic chemistry first assembled itself, one slow reaction at a time, across the frozen surface of a microscopic dust grain. And the basic energetic trick every one of your own cells still uses to stay alive, pushing charged particles across a membrane to generate usable energy, is, as far as the best current evidence suggests, essentially the same trick life first learned inside a mineral chamber on an ocean floor that vanished into the planet's own restless geology long before anything resembling a continent as you'd recognize one today ever existed. Thank you for spending this long, quiet evening with us here at Pale Blue Drift, tracing the deepest, most ancient roots of everything currently alive on this planet, all the way back past contested rock formations, past reconstructed ancient genomes, past hydrothermal vents on an ocean floor that no longer exists in any recognizable form, and further still, into the patient, silent chemistry of the space between stars. If slow, deep mysteries like this one, reaching back far enough to make an entire human lifetime feel almost impossibly brief by comparison, are the kind of thing that helps your own mind settle into something calm and curious before sleep, there will be more evenings like this one waiting for you, more ancient threads to follow, more quiet, unhurried nights built for exactly this kind of stillness. For now, let the rest of tonight be simple. Whatever ancient chemistry first found its way into a mineral chamber on an early ocean floor 4 billion years ago has already done its work, patiently, thoroughly, across every generation since, all the way down to you, resting exactly where you are right now. Nothing more needs to happen tonight. The story that began before this planet even existed has already arrived, safely, completely, in the quiet, steady rhythm of your own breathing. Sleep well,